So thank you all so much for joining us this afternoon for our Knowledge Portal Network webinar. I'm going to go through some updates from the KPN, including our various events, and then I'll hand it over to our featured speakers. So coming up, we have the American Diabetes Association conference that'll be held June 20th through 23rd in Chicago. Please feel free to come visit us at booth 948. We'll be in the NIDDK corner representing the Common Metabolic Diseases Knowledge Portal, and we'll be unveiling a brand new portal, the Mammalian Adipose Tissue Knowledge Portal, which will be on the first day of the event. But if you're going to be at ADA before, so on Friday, June 20th, Julie Jurgens is going to be presenting on this brand new portal during our session, the linking mechanisms linking adipose health with lipid and glucose homeostasis. And this is going to be an oral presentation in the Convention Center West building. So if you're available, come please listen to Julie introduce the MAT-KP for the first time. We are really excited about this event. There's going to be more upcoming in the Knowledge Portal Network with MAT-KP, but we're just very excited to have this as our first ever entree to this portal. And if you're going to be there right after Julie's talk, we're going to be having a NIDDK networking reception that'll be to celebrate the start of the conference. And as I said, this will be right after Julie's talk. So this is 6:30 to 8:30 in the exhibit hall at the NIDDK Science Corner. These are the various organizations along with us who will be a part of this. And we're really looking forward to having everyone there. And on Saturday, we're also going to be doing a celebration as we are going to be attending our 10th ADA this year as CMDKP. We're going to have an ancillary session with some scientific talks and networking. And this will be, as I said, Saturday, June 21st, 6 p.m. to 7:30 p.m. This will be at the Hyatt Regency McCormick Place in Grant Park D. I'll be sending more information regarding all of these events to our mailing list, but if you're going to be attending ADA, please let me know. I would love to schedule some time to meet with various scientists and others that are going to be at the booth. We'll also have some demos throughout the week, and we'll be spreading that information on our social media and listservs, but we're very excited for another year at ADA. And looking even further into the year, we will always be attending ASHG. So with that being said, we're going to be there October in Boston, and we'll be showcasing both the CMDKP and the A2FKP. I will be sharing more information as we get closer to, but we're really excited about this event, especially being in Boston. We're going to plan a few other networking events since it's in our home turf. We'll be able to do a little bit more, so please be on the lookout for that as well. One other webinar series that I just want to showcase is our Common Fund Data Ecosystem public webinar series. And our next session is going to be Friday, June 27th from 1 to 2 p.m. And we'll feature Andreas Bucco from HubMap, and he's going to give a presentation on multi-scale virtual reality exploration of the 3D Human Reference Atlas, proving novel insights into health, aging, and disease. So we look forward to having that event being hosted soon as well. Just some upcoming dates for our various meetings. The next is going to be July 10th, which will be a focus group. And our next webinar is going to be in August. So we look forward to having you there as well. And so with that, I will hand it over to our featured speaker, Mike Love, who will be giving some updates from the IGVF working group. And then we'll have a demonstration of the portal that we've collaborated on from Trang from our team. And just to note, we are going to have the chat available, and any question and answers, we can address those as well. So with that being said, I'll hand it over to you, Mike. Thank you so much.
Great. Thanks so much, Mackenzie, and thanks for inviting us to come present. I'd hope that let people feel free to ask questions throughout. I'll be checking the chat. And yeah, it would be helpful to have more of a conversation. So let me throw up some slides. Okay. And oh, give me
That
one.
looks great.
Okay. I'm gonna go back to window view for me so that I can also see the chat. Uh, great. Okay. So now I can see the chat as well. Uh, okay. So thanks again. I'm here to give an overview of this consortium, so the IGVF consortium and the data and like how we're making the data accessible. And I start off with the credits. So I basically pulled together a lot of slides from various different teams and I want to just mention, a few of the names of the people where I'm borrowing from. So there's a working group within IGVF, which I'll define. I'll define the consortium in a minute. But we have a working group devoted to this thing called the catalog, which is kind of like the way that we make our data accessible to the broader community. And there are two key data centers. So they're actually DACCs, Data Analysis and Coordinating Centers, WashU and Stanford. I'm showing work from both of those. So like on the catalog working group, we have Daofeng Li and Ben Hitz, and also Jesse Eingritz has done a lot of lead work on putting together catalog visualizations and tables and things like that. Also, there's been IGVF groups and early adopters of kind of pushing our data out. So one of those is the coding group within IGVF. And another one is the Lipids Knowledge Portal project, which has been really excited to see come along. And you'll hear more about this after I'm done. Um, and also, so all IGVF consortium members and affiliates have kind of contributed to what I'm going to talk about today. And finally, Martin Morgan's outside of the consortium, but helped on creating an R package for looking at the data. I'll show that at the end. So, just like where we are orienting yourself to this consortium, it's an NHGRI consortium which began in the like late part of 2021, and so we're in heavy into production and looking towards the end of the first round of the consortium. And so the kind of key outcome of the consortium is going to be some resources, so data I'll talk about in one slide, as well as tools and models um, and hopefully also biological insights into variants and genome function and disease. So there's a description, a perspective paper, kind of announcing the consortium, and I just highlight and I'll read because why should I do better than this nice paragraph here? So the consortium IGVF stands for Impact of Genomic Variation on Function. And the goals is to combine approaches in single cell mapping, genomic perturbation, predictive modeling to investigate relationships among genetic variation, genome function, and phenotypes. So it's very broadly defined, creating maps of across hundreds of cell types and states, describing coding variants and how they alter protein, as well as non-coding variants changing regulation of gene expression, and then how those propagate through networks. Um, so there's both experimental data groups as well as computational groups of people building predictive modeling, sequence to function models, and we also put out standards and pipelines. So that's a paragraph I wanted to just read through, but we also have kind of more diagrams. The idea of IGVF as a resource to the community would be you can look up a variant and quickly and easily find relevant functional data, computational predictions and relevant methods. And we kind of conceive of a broad base of user groups. So it could be a genetics researcher who's interested in studying, say, variants that relate to type 2 diabetes and wants to see what kind of functional data there are. It could be computational methods developers that want to, use data to benchmark their models, or experimental researchers who want to design a study. So kind of analogous to, I think, a nice analogy is the ENCODE consortium and how valuable that has been for all of these kind of use cases. Um, so to give a picture of like what are these perturbations we're talking about? So we kind of conceived of a framework for these experiments as mapping, perturbation, and prediction. So on the mapping side, there are, a lot of single cell mapping efforts within IGVF. Um, so kind of you can see in small gray font the assays, and in blue is kind of uh, the what the goal of this would provide for the next step in the framework. So single cell mapping lets us know about gene expression and chromatin accessibility in individual cell types that are relevant. Um, Genomic perturbations such as base editing, prime editing, reporter assays, CRISPR, AI, um, as well as this is kind of the non-coding side on the left, and on the coding side, like VAMP-seq and cell sorting, cell painting, etc. The idea is to perturb variants, whether non-coding or coding, elements and genes, and measure effects. Um, and those feed to help train and evaluate predictive models. So there's also a number of groups coming at the data from the predictive modeling side. And so the idea is that the modeling would help better design second round of mapping and perturbations, and the perturbations help improve the models. The maps help prioritize which variants to perturb, etc. Just checking. Okay, see if anybody's asking questions. Please feel free to jump in at any time. Um, so there's kind of focus areas, like if you, from the variant perspective, you know, which missense variants of unknown significance are pathogenic. From the non-coding side, like which variants affect the expression of which genes. It's very broad. There's, I think, 21 centers funded, and so it's across many different systems and samples and like assay types. So how does it come all together to be available as a data resource? So there's the IGVF data portal.
Mike, can I have ask a question?
Please.
Can you go back to the previous slide? So on the left, right, like this is kind of a framework of all the data types that you have, and it's almost like a feedback loop between the different data types. So I'm wondering, conceptually, how does that work? As in, do you start with the initial data set and then you kind of start building them up and then kind of input that into the next step, or just overall, like how is the
Yeah,
process?
I can speak to that question just as part of a center in the bigger consortium. So like we designed at UNC, we have an MPRA where we were interested in in looking at the allelic effect of variants and associated with some GWAS traits of interest. And so we said these are some variants of interest, had a library design step. And then we asked predictive modeling groups, we reached out to a number of them saying, like, before we do the experiment, what do you think about these variants? Like, which would you prioritize? And so a number of groups gave us feedback, and like we had the goal to kind of comprehensively look at their allelic effects of all of them first. So we didn't filter based on those, but now that we have the data back, we can look to see, oh, like which annotations were most helpful in highlighting where we see empirically an effect, or maybe where another way to use kind of predictive annotation would be to say, can a predictive model help us see where our assay agrees with another assay? So maybe like each of these assays has their own, sensitivity and specificity. So like, can predictive models help us zero in on like, where's the agreement or where's the biological relevance? Where do we best line up with like data from human populations? That's just one step of the cycle, but like basically it happens in conversations in working groups and focus groups. So in designing experiments and in the computational modeling groups, it happens like what data sets do they use to train and evaluate their models?
Yeah, thank you.
There's a structure in the data portal where you can see which models were trained on which data. Like that's also an interesting aspect of the consortium, is like you have to keep track of all this, right? You don't want to end up evaluating a model that was trained on somehow there was like a data set that was also used in training. So, even keeping track of all of that is very important. So that's a nice segue for me to introduce the data portal. There are data at the level of raw data sets, processed data sets, and also the way that the data is modeled is kind of more complex because the data sets themselves are in a way more complex than, say, like the ENCODE project previously. And so I'm going to show you I'm going to click essentially on this process data sets and also on the schemas just to give you a picture of what does the data look like in the data portal. Um, so like I just pulled this up yesterday and grabbed the number of processed data sets these are not released numbers, but these are kind of data that you can see within the consortium, and then these data sets eventually become released. And so once they pass like quality control and all the metadata is correctly annotated, um, so like this is just, seeing what's up there right now that you could that is being uploaded. Um, in terms of the schema, it's more sophisticated, kind of handles more complex representation of data. So you can see there's analysis sets, um, information about libraries that went like, for example, you design a library and that gets integrated into some cells or samples. Um, Various different modifications, phenotypes, everything is kind of tied to ontologies to make it better for predictive modelers, or for people trying to annotate, variants and do this in a programmatic way. Um, This is a picture of the data portal, like some screenshots that I took of a data set. So this is that I just used our own data set from UNC. So this is, a data set where you have up here a uh, the measurement that contains the data. Um, you have information about what the sequences are, why were these variants tested? So rich metadata about the variants that went into a cell. And then you have the results, and all the results are nicely associated with the original file so that you could go actually back and reanalyze it a different way. So this is providing kind of the computational reproducibility. Yeah, this is just a schematic that I made. This is the schematic of the linkage of the data that I showed in the previous slide. So like, how do all these things connect to each other? So again, I think it's from my perspective, it's very rich metadata, allowing you to kind of traverse all the information. And this is just for one type of experiment. This is for a reporter assay, massively parallel reporter assay. This would look totally different if somebody were doing a CRISPR screen or single cell mapping. It would all have kind of a different structure to the data. Um, so that's the data where the data lives in raw and processed form, and you can go download from that site. There's also a catalog, which is a more user-focused deliverable. And so the idea is that all of these assays that I described, so the experimental assays are these purple lines. Um, all of these assays are helping inform genome function. And the idea from the design of the catalog kind of early on in the consortium was to use a knowledge graph. So rather than have all the data be tied to assays, which, like assays come and go, DNA-seq is used to measure chromatin accessibility, and then comes ATAC-seq, and like rather than have everything be tied to assays, the user-facing side of the data is tied to biological entities, which maybe have more permanence. So like on the catalog side, you can look up a gene and you can look up regulatory elements and cell states, etc. So I'll show you, it's referred to, this style of representing the data is referred to as a knowledge graph. So let's see. Oh yeah, here I have a picture of the knowledge graph. So if you go to the catalog website, you can look up variants and genes and enhancers, and pull down basically the processed data from the data portal. So this is kind of once the data is processed and kind of ingested into we're tying empirical observations and predictions to biological entities of interest, it goes into basically a graph that you can traverse. You can traverse it like on a website, or through what we call different apps. And one of the apps would be what Trang's going to show you in a minute. You can also look at it like as a graph. And you can also access this through an API. So you can, programmatically walk around the graph and pull down empirical or predictions. I have one slide here from Minji Lee from the Stanford DACC, which I thought was super cool. So Minji's been working on translating natural language queries into query language. So the data is stored in a Rango database, and so translating a natural language query into the appropriate query language to query the database is in progress, and it seems to work pretty well. So this is like a really exciting and promising way that people could interface with the IGVF data. Work in progress. I'm gonna just quickly show a couple slides about, I worked on a R package, actually a bioconductor package, which is now in the latest release of Bioconductor, which allows you to access the data from R. So if you want to find, for example, genes and so you have a query gene and you want to find variants that have been associated with it, either functionally, like through an empirical data point or through a predictive model. You can use the gene variance function, and then there's other functions. We haven't written out all the edges in the graph, but we just kind of started to prototype, would this be useful to allow this kind of access? So yeah, so there's, various ways. It's basically just a wrapper to the API, which you could also call from Python, for example. We have Python notebooks showing how to access the API. You can use various different identifiers. You can filter, specify like log10 p-value, etc. So in this R package, you pull down the data and like it can be represented in native bioconductor types. So if you ask for elements overlapping a certain range of the genome, you get it back as the appropriate type of object in bioconductor, which is like called a genomic range object. And it has metadata, for example, like which experiment detected this element or detected this link from a variant to a gene. So this is my last slide. A lot of what I showed you today is a work in progress. As the data's been coming along, all these groups that I showed the work of have been trying to figure out how to visualize the data, how to make it accessible. We're also doing outreach to user communities. We're very interested to know how it's useful to genetics researchers, even clinical geneticists, working on the API. And so optimizing for speed, anticipating what will typical users be asking for, figuring out the database load that will be typical, defining data formats and making these very easy to find. So if you're looking at the catalog, you should be able to go back all the way to the data portal and download a file, like a flat file. Figuring out the balance between the knowledge graph and the data portal. So filtering and prioritization of results is still, I think, an important question. And there's also, I didn't mention today, just due to lack of time, there's also an integrative analysis step that's going on. So there's groups that are kind of coming together and trying to figure out like, what's the important biological story? That's being done by lots of specific working groups. So for example, like the coding working group, all the groups that are assaying coding variation come together and try to figure out like what together can we do with all the data we've generated. So I will now hand off to Trang, who has a more exciting demo, a live demo or demo of what she and others have been working on.
Thank you, Mike. Should we take a second to see if anyone has any questions?
If you have a question and you would like to ask it, you could raise your hand and I can take you off of mute. Or if you want to ask anything in the question and answer or chat, we are still monitoring those as well.
I guess I have a question for you again, Mike. So you guys have kind of generated a lot of different data types to understand variant function. And I'm curious, what are the main kind of users or target audience of all of these data, and how would they reach out to you to get certain information to answer their research questions?
It's a great question. We went through a process like in year 2, I think, where we had all the groups that were, all the different focus groups and working groups try to define what are called user stories, like write out, who is your user, who's the user, and what do they want, and what do they do, what are they trying to do? And so it's very diverse because like the assays are so diverse. So some groups are measuring cell type specific gene expression and chromatin accessibility. That's very useful, for a particular group. This other group is measuring, doing saturation mutagenesis of every amino acid in many, hundreds and thousands of proteins. So those are both kind of comprehensive ways of assaying, but their audiences are very different. Where like the coding variation audience, they have close ties to like the clinical genetics groups, and they have appropriate ways of making that data available and accessible to groups that would use it as functional data points in variant interpretation. But that's a very formal process. And whereas other data sets from IGVF kind of, I think probably have more of a, some of them have like very clear user case of computational biologists trying to help someone design an experiment. So it's really broad. But each group kind of, they have to do their own outreach because like it wouldn't make sense for a non-coding person to try to figure out the use case for a coding center. So we've been pushing, each group does its own outreach to the relevant user communities.
Thank you. I think we have a question.
Great. Will only data from members of the consortium be included in the portal? No. So I didn't mention this, but actually right now the majority of the data is from previous existing consortia. So for example, like just to name one thing, like all, GTEx EQTLs, a lot of external data sets have been ingested. Ben Hitz has been the primary person doing that, but that just makes it very easy. You don't have to go to another location to like make use of that data. So like encode CCREs. Yeah, I have a slide. I can go look at all the different major data sets from other previous efforts that have been included here. Good question.
All right. Thank you, Mike. I'm gonna kind of move over to my demo then. And in the meantime, if you guys have any other questions, feel free to drop in the chat, or also interrupt me, whether it's for Mike or for me, regarding the lipids KP. Okay. Can you see my screen, the slides?
Yep.
Okay, great. I'm just gonna turn off my camera because I'm looking at the second screen and I don't want you guys to have to look at the left side of my face for the entire 20
You're
minutes.
fine, Trang.
Okay. Well, maybe this is fine. Share my screen. Hi everyone, my name is Trang, and I'm a computational associate at the Knowledge Portal Network, and I've been with the team for the past 4 years, and also the past 2 years I've kind of been working with Mike, Eric, and Kusha, and a lot of other members from the IGVF consortium to kind of develop one of these prototypes that could bring out the data from IGVF to the broader community from a disease perspective. So as Mike mentioned before, IGVF consortium is trying to really study and understand the variant function from different perspectives. And one of them is from a human disease perspective. And it happens to be that, our Knowledge Portal network main focus is also to understand complex diseases from a genetic and genomic perspective. So it kind of is a very nice collaboration between the two groups, I would say. And it's been my joy working with you guys at the IGVF, and I'm excited to demonstrate the portal live today, because last year I also did this demonstration with Eric Van Buren, but we had to use the slides instead of the live demo, just because of, just to be safe technically. So before I kind of start the demo, I'm going to go over a couple of data types that we've ingested from the IGVF consortium. So Mike kind of showed lots of different data types, and it's not really it's not always very straightforward to kind of answer this question of, so how do we combine all of these data types to answer a specific question? And so we want to kind of combine these in a way that makes sense visually and also scientifically. The first kind of data that we got from IGVF was functional genomic data. So for example, we have cis-structured elements or CCRE annotations from ENCODE, as Mike mentioned, because they did kind of ingest data types from public resources and also from their own projects as well. So this is one data type. The second one is, in terms of functional genomic data, is variant to gene links that was generated using data from Hi-C loops, CRISPR screen, or co-localization from different data sets, primarily in HepG2 cell types and liver. And the reason why we're kind of focused on liver right now is because the lipids KP is kind of the prototype portal that we're building, and livers happen to be one of the tissues that are super relevant to lipids. And the final, or the third, I would say, not final, type of data is region to gene links. So all of this is kind of to connect the different peaks of the functional genomic regions to actual genes, so we can understand better what regions are affecting the function of certain genes. And this is from the 10x liver multiome data, using lots of different methods, also from IGVF labs. And all of this is just the scaffolding data that we have right now. And in the future, we're hoping to get more data into the portal. So that is functional genomic. The second pieces of data is genetic association data sets, because again, we want to look at the variant function from a disease perspective. So all of these familiar types of genetic data, for example, GWAS summary statistics, we have the GWAS from the GLGC consortium currently for 5 lipid traits across 6 different ancestries to start with. And we also have fine mapping results from the same study as well. And finally, we have bioinformatic method results. So all of this is kind of second level. I think I have a couple of questions in the chat.
There is one question
Oh.
that is asking, what additional data would you aim to collect to supplement the scaffolding data?
Yes, great question. So we are mostly kind of primarily using the data from IGVF consortium, and they do have, and correct me if I'm wrong, Mike, they do have an API that kind of help us query data directly from IGVF. But so in the future, we're going to establish a system where we can just directly get the data from them. Because for now, all the scaffolding data is ingested manually. So like I get the data and then I upload it to the lipids portal. But yeah, hopefully most of the data types that are compatible with the lipids portal's features will just be ingested via an API or something like that.
And
Thanks,
you
Julie.
know, sorry.
No, you're good. I was just saying thank you.
The API is also, really not that hard to try out. Like I'm not a big database person, but like it wasn't too hard to, to build from it, and it's accessible, for example, from Python. So it's pretty easy to query from the API.
Thank you. Okay. Yeah, so the final piece of and thank you for the question, Victoria. The final piece of data is the results from bioinformatic methods. So for example, we have the consensus variant to function scores, or CV2F, in multiple cell types and tissues that were developed by members at Kosude lab. And basically they use, I think, a novel method to combine different data from GWAS or EQTL or functional genomic annotations to come up with a set of probability scores for a variant function related to a certain cell type or tissue. The second type of annotations, results from bioinformatic methods is favor annotations that were developed by favor camp focus group, also from IGVF. And we have Eric Van Buren today with us actually to answer any questions you might have about this data. But basically, they generate different integrated annotation scores for billions of variants, both coding and non-coding, using the pre-existing annotation that we already have from public database, for example, like DB and SFP. And another kind of results we have is from a perspective of a phenotype. So because we have genetic data, we also have results for genetic correlations between different phenotypes and global enrichment of genomic annotations that we generated using LD score regression methods. So with that being said, I would like to, before I do the demonstration, I would like to thank members from both teams, from both the Knowledge Portal Network and IGVF that have made this happen. So I'm going to try to do a live demo today. Okay. Actually, it's half live because I kind of preloaded the web pages to make sure they work. So, so this is the kind of and again, everything is due in development. So if you have any questions, feedback, comments, please let us know so we could improve the site furthermore. So the first thing that you're going to see is just the homepage. And on this homepage, you can see all of the main pages to the top right. So for example, we have the homepage, we have the phenotype page, signal sifter, variant to function, and finding documentation. So documentation is pretty obvious. If you click on the documentation pages, you could see the information about different data types and different methods that we have. And we're always working to improve the documentation to provide more information. But the three main pages really are the phenotype page, signal sifter, and variant to function. So I'm going to start with the variant page or the variant to function page. From the start of the page, if you input a variant ID, you're going to see lots of different sections. And the first one is the PheWAS plot. So this is the PheWAS plot that we kind of imported from our main portals, where we kind of show the genetic association between this particular variant with all the 1,000 phenotypes that we have on the portal. And you could filter by the p-value. So if you want to restrict the p-value to only less than 0.01, and you can do that, and you can see the different phenotypes that are nominally significantly associated with this variant. And as you scroll down, you're going to see lots of different functional information about this variant. So the first thing is the favor scores, as I mentioned before, developed by favor team. So the first two sections of favor are kind of what I think you can get from public resources like DB and SFP or the two VEP. So you would see the generic information about this variant, allele frequency, gene code, category, and the different score from different annotations in this table. So if you kind of just click on the column annotation, you could sort the annotations and you can see the score that was generated for this variant. But the novel thing about favor is that not only it gives you the individual scores, which sometimes could be very overwhelming, but favor developed different algorithm to integrate all of these scores, kind of categorize them into different groups of annotation. So for example, you have conservation score, or you have epigenetic score, or you have protein function scores, and all of this is on the same Phred scale, and they kind of integrate the different annotations from different algorithms to give you one kind of consensus score for this variant. And again, as you scroll down for this table, you can also sort by different columns in here, whenever you click on a different column. So that's favor scores. the next kind of scores that we can learn about this variant is variant to function score. So this is developed again, as mentioned before, by a day lab at IGVF, and it also shows you a plot that has the different scores from different cell types or tissues or different data types that you can also see at the table below, and you can sort and filter. So for example, if you want to look at the variant to function scores for this variant from blood, you can see that the first column here is blood, and it also has the specific biosample that this data was generated from, as well as the V2F scores of 0.36. So this is on the scale of probability. So from 0 to 1, the higher, the more likely that we're sure that this variant is having an impact on this tissue or cell type. So that's kind of like what the information we have for now for variants on the variant page. Do you have any questions for me so far before I move on? Okay, great. So that is the first page, the variant page. I really like this page because it kind of gives you an overview of what a function is doing in the genome, but it doesn't exactly give you more detailed information about this variant in relation to the gene or the region around it, or a specific phenotype. So to do that, we're going to transition to the signal sifter page that will give you a much in-depth view into the variant and the region around it. So the signal sifter allows you to look into both the genetic associations and the functional genomic annotations for a particular phenotype. In this case, I chose LDL, low density, sorry, I blanked, LDL cholesterol for European ancestry. So the first section that you see for this phenotype and in this ancestry is genetic associations, as you normally would see in a lot of our pages on the main portals, other portals outside of IGVF. So you're going to see all the variants associated with this phenotype in this region. I set it as LDLR as I think as a main gene for this region. So you can see the local zoom plots and the table that also has the same data. And over here on top, you can filter by variant ID, RSID, or lots of different columns to find the signal that you're interested in. So that is the first plot. The second tab that I want to highlight in this section is also the global associations from a clumping perspective. So lots of the variants are in LD with each other, and sometimes we don't really know which one is driving the signal, and we don't want to kind of count the two variants to be two different signals if really they're the same, right? So this kind of tab would show you across the entire genome, if you clump all the variants in LD with each other, how many signals are you really seeing? So for example, the first row right here, you only see one variant, which is the lead variant, and then on the 1, 2, 3, ninth something column, you could see the clump region that this variant is the lead SNP in. And when you click on it and you click set region, it's going to replace LDLR moving forward as the region that we're focusing on. And a very similar approach is from a gene perspective. So if you click on the final tab, you can see the same thing, but instead of a clumping region, you're going to see the gene results that I believe from Magma that we implemented, also using all the GWAS information we have for this particular gene. And again, you can sort and filter by different columns here, or you can just hover on each of these dots on the plot and see the information about a certain gene as you see fit. And you can also, again, set the region that will kind of inform the rest of the page. So right now I'm still leaving the region as LDOR. So that is the genetic association section. The next section we can see on this page is the credible set. So again, this is also calculated from the GWAS association. Right now it's showing the credible sets that we pre-computed like within our own software. And you can see all the credible sets ID, the region of these credible sets, And if you click on one credible set, it's going to lead you to the next section, which is the credible variants within this credible set. So for example, I click on the 415 credible set data set, and it leads me to all the credible variants in this set. And you can see that there are different information, for example, the variant ID, and the most important thing is the possible probability of inclusion. It's actually PEEP and not PPO, probably a misspelling. And you can sort and filter on the variant to see which variant has the highest probability of being causal to this phenotype in this region. So that's the second section. Any questions so far?
I have a practical question about the gene labeling. So it's always nearest. What if, we learn about the locus and we have some other data to show that it's not the nearest gene? Like, is there a way to kind of override nearest?
Which section are you talking about?
And when you label variants, do you label nearest gene
Um,
in the associated variants?
oh, I see. Oh yeah, so this is are you talking about
Oh,
the
maybe I hallucinated it. I
so
thought there
we
was
do
somewhere where you had nearest gene for region for like a GWAS signal, but I maybe made that up. So then that answers the question. It doesn't.
yeah, so all of this is from the region perspective. So if you have a
Got it.
variant that's, nearer to LDLR, but it's really actually also near to SMARC
Got
and
it.
4,
Great.
right? I can see all the other genes. Yeah.
Thanks.
No, thank you. Where was I? Oh, the next section. So the next section is kind of the results from different bioinformatics methods that kind of summarize the phenotype from the functional genomic perspective as well. So the first thing you see is partitioned heritability. So this is the results from LD score regression that kind of tells you in all of the GWAS signal that you're seeing, which one of these functional regions are contributing the most. I think essentially that's what it's saying. So we get the annotations for liver, the CCRE annotations for liver from ENCODE, I believe. And you can see here in the plot just the overall results, so the p-value, the log p-value, and the fold enrichment fold for each of these type of annotations for this phenotype. So for example, in this case, I think we can see that the pink dot over here, which is DELS, I believe it's like in deletions regions that is most enriched for this phenotype using this annotation. And as you click through the different tabs, you can kind of see the same information, but from different perspectives. So for example, I think this is broken down by the GWAS from different ancestries. And then when you click on the tissue annotation, level annotations, you can see exactly the tracks that show this data. So when you hover on these tracks, you can see the different regions within this, around this LDLR gene that has the active annotations for liver. And I believe biosample is just on the level of biosample, and it's still liver in this case. So that is kind of one way that we integrate both the genetic associations data that we have and the functional genomics that we got from IGVF to summarize what we know about this phenotype. And the final piece of data on this page that I want to show you is the target gene links and the variant to gene links data. So the first one is basically the linkages between a gene and a regulatory regions from different methods. Again, I also got this data from IGVF, Mike. And you can see which regions are kind of affecting the function of particular gene. And the second tab is the SNP to gene links, which is telling us which genes are being linked to the variant, like the variant in this region. So I want to show you something that I forgot to show you earlier that was super important for this section, is that if you see, can you see the vertical line here? It's pretty faint, but I hope you can
Yep.
see. So throughout the entire page, all the tracks that we're showing are on the same scale and on the same like coordinate plot. So if you scroll up and you see a signal here in this local zoom plot, and you see, oh, I'm interested in this variant. I want to know more about this variant, right? So, but if you have to scroll all the way down, you kind of losing what the variant is. So this is one way that we kind of keep track of the variant. So if you click on the variant, you kind of star it, it will show a vertical line, and you can just scroll all the way down to the page and see whether or not this variant falls into whether a functional regions of these genes, or it is linked to this particular gene by any method. So it's kind of, I think it's a very nice way to kind of dissect the signal, a certain signal from a different perspective, right? So you have the genetic pieces and you have the genomic pieces from different data sets. Any questions so far?
What if you had, I'm just like thinking out loud, if you had a variant with 100 variants in LD 0.99,
Yeah.
maybe it may be useful to kind of highlight all of those. Sometimes you're really in trouble with the GWAS and like,
Yeah.
well, you could guess, but yeah.
Right, exactly. I think I mean, that's what we're here for, right? Spending decades of our lives trying to pinpoint the causal variants. But I agree with you that it's really it's not a really trivial question to answer. Which one is exactly the one that's driving the signal? And you can see right here in LDLR already lots of different variants in this region, and you kind of don't really know which one is causal. So I think it's important to again put the multiple pieces together. So if you click on these different variants, the chances is that not all variants are going to fall into all of these regions. So you kind of can filter down the variant that way using different data types.
Great, thank you.
Okay, and then
There's a question, Trang, in Q&A. Oh no, is Trung frozen?
For me, yes.
Okay, well, maybe Mike, you might be able to answer this.
Yeah. I don't know about
for this phenotype.
Oh,
sorry, Trung, you froze.
we lost you for a minute. Yeah.
Am I back?
You're back.
Okay, great.
There's a question about if you can upload your own GWAS summary stat data.
Yes. So we're always happy to take user contributed GWAS data. So this is in terms of the knowledge portal network, yes. But because this is also a portal that we're developing with IGVF, so I think that question I would kind of defer to Mike.
Oh yeah. So like I posted in the chat, they ingested quite a lot of data sets. So, but like yeah, if you wanted to visualize your data in general with IGVF data, I mean, well, yeah, I think it depends on the app. So we're called like this Lipids Knowledge Portal is an example of one we call like app, one way of interfacing with the data sets, and there's many others. And so, lots of people are thinking of ways to visualize their particular data. So it kind of depends on each one.
Yeah, but I think the short answer is yes, right, Mike? That we're happy to take data from the community. Yeah, thank you. Yeah, so sorry, back to the phenotype page. We're just seeing a kind of summary of genetic associations for LDL cholesterol. This is the top, I believe, 1,000 something, 704 variants that are associated with LDLR. And as you scroll down the page, you can also see the gene level associations that you already saw on the signal shifter page that I just showed you, but kind of just a simplified version of it on the phenotype page. And the next thing you see is the pathway associations, also from MAGMA with this phenotype. So you can see the different pathways, for example, plasma lipoprotein here associated with LDLR cholesterol, or coronary artery disease, atherosclerosis, which makes a lot of sense. And then the final section you can see, actually not final, the final two sections are the results from LD score regressions. So the first one is genetic correlation with other phenotypes. So it makes sense that a lot of these top phenotypes that are so genetically related with LDLR are a bunch of cholesterol lipids traits. And then finally, the enrichment from LDSC that is similar to what I've just shown in the signal sifter. So yeah, that's basically all the data types and features that we have on Lipids KP for now, and hopefully in the future we'll incorporate more data from the API directly, and so we could expand the portals further. Thank you. Have you take any questions, comments?
It looks really cool. Great work, Trang and
Thank
team.
you. It's teamwork, yes.
Thank you for all your collaboration and help on this. We wouldn't be able to do all of this without your team. So thank you, and thank you so much for taking the time today. I think it was a really great presentation, and we appreciate having you here. Before we log off, does anyone have any questions? If you want to answer any live, just raise your hand and I can take you off mute. Or you can use the chat and question and answer. Okay, I don't see any questions, but thank you all so much for joining us today. Just a reminder, our next session is going to be July 10th, and we'll have a focus group meeting. So thank you again to everyone for joining, and for Mike and Trang for presenting, and we appreciate having you here. So thank you, and have a great rest of your afternoon.
Thank you. Thanks, everyone.
Bye.